This is episode 1 of 3 of the Immunology Podcast, Immunophysiology with Dr. Henrique Veiga-Fernandes. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Rout. Welcome back to the Immunology Podcast, where we have conversations with immunologists. The Immunology Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the Immunology Podcast, rate us and leave us a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today we have Dr. Henrique Veiga-Fernandes for the Champalimaud Foundation on the podcast to talk about his research on crosstalk between neurons and the immune system. We've also got our usual roundup of recent highlights in immunology news coming up. But first...
Can you believe it, Jason? We're less than 2 weeks away from Immunology 2025. If you're joining, check out the program and plan your meeting at immunology2025.ai.org. And we're so excited because we're going to be hosting a live podcast show this year, like last year. And you can join us in room 317A and B at 10 a.m. Hawaiian time on May 5th. We'll be reflecting on over 100 conversations with immunologists over the past 4 years that we've been doing the podcast. And we really hope to see all of you there. It's going to be really fun and a great chance to hear about 100 episodes of the Immunology Podcast.
I am going to be wearing a ridiculous Hawaiian shirt.
As is your right, Jason. Indeed.
Well, it's not on May 4th, so I'm not wearing a Star Wars outfit this time again. It's on May 5th.
That is true. That is true. That was last year. You wanted to wear your Jedi costume, but May 5th is a Liberation Day here in Europe. So maybe
liberation from which war?
World War II, the end of World War II. At least here in the Netherlands, I'm not sure. I think it's I think it's like the Netherlands specific.
Probably, because it didn't all happen in a day.
Yeah, exactly. I think this is Liberation Day here in the Netherlands. So people are very, celebrating another year in which fascism was defeated.
D-Day and Memorial Day and all those things coming from the U.S. that we celebrate.
Yeah. So anyway, are you excited? I'm so excited.
For Hawaii? So I guess I can share this here. We'll share it live too. But the hotel we're going to is a special hotel for me. One, it's where my parents had their honeymoon. It's also where at the age of 2, I ripped my hand from my parents' arms and said, "Look, Mommy, Daddy, I know how to swim," and ran into the ocean.
That sounds fun for your parents.
I had never swam before, but I'd watched people swim.
So you thought, how hard could it possibly be, Jason of 2 years old?
And I swam.
Nice. You swam how far before your parents?
Not very far.
How did that happen? It's like not like you could have outrun them with your little, 2-year-old legs.
I was close to the ocean and I booked it.
Well, I'm glad you were not fast enough to, get swallowed up by the ocean.
The pools there, which is there's 2 side-by-side pools that overlook the ocean that you'll see, is also my first memory that I actually retain. So in a way, the place is super special to me. My martial arts instructor is on the islands. Hopefully I'll go see him. I'm going to take a class one night. So if you see me with a black eye the next day, there's a reason, things like that.
All right. Okay. Good to know. So we're all going to be experiencing some fun stuff then. I just hope that the few days I'm going to go earlier are enough to get over the massive jet lag. I'm gonna be.
Isn't it 12 hours for you? It's just complete inversion.
It's like literally on the other side of the earth. It's just a bit. It's okay. It's gonna be fine. I have faith in myself. Anyways.
All right.
So
while we've been discussing the potentiality for muscle-induced locking and weakness while sitting on a plane, I'm going to discuss to you the muscle acetylcholine receptor and immunology related to myasthenia gravis. And if that wasn't a segue, I don't know what is.
Brilliant. You always get me. I'm sure our listeners really enjoy your segues and, are taking notes.
They should give feedback on how bad they are.
Please. Please do give feedback.
Save Brenda.
Find us on Blue Sky at immunologypodcast.com and make sure to let Jason know how good his segues are, because I think the man needs to know. Anyway, so you're saying Myasthenia gravis, please go for it.
In Cell, we have autoimmune mechanisms elucidated through muscle acetylcholine receptor structures. First author is Huan Li, and corresponding authors are Colleen M. Novello, it's not clear, and Ryan E. Hibbs. So this came out on May 1st, which is in the future. It's online now. You know how it goes. So myasthenia gravis, right? It's antibodies to the acetylcholine receptor. So now you can't have acetylcholine signal to your muscles, so you get muscle weakness, and you can then have like lung problems and breathing and die. So you have to use immunosuppression to stop this. The paper is going to be pretty quick to describe because this is a structure paper, and you just got to go look at the pictures. But they are the first group to get a structure of the human acetylcholine receptor. No one did it before, which is just, of all the things I'm like, I thought we didn't have, I didn't think we didn't have that. So the problem is they have improper assembly of the receptor at scale, apparently has been the problem. So they overcame this by using an affinity tag to the delta subunit and then pairing alpha 1 delta and beta 1 epsilon units together to minimize assembly. So they also did this, and what's interesting about it is this is how it's arranged on the chromosome, where alpha 1 and delta are on chromosome 2 and beta 1 and epsilon are on chromosome 17. So it actually mirrors the human assembly, at least in terms of this receptor is on multiple chromosomes, which is weird, right? The subunits are on completely different genetic chromosomes. What? That's not typical for genes or for proteins, right? To have subunits on different chromosomes. But in this case, they are. And it generated stable cell lines that prove through electrophysiology, right? Because it's an ion channel, that they made the receptor. And then they cryo-EM'd it. And then they cryo-EM'd it with like 9 antibodies from myasthenia gravis patients that all had different known function. So you could have antibodies that block the acetylcholine from binding. You could have antibodies that cause internalization, complement, mediated killing, all this stuff. And they then look at where all these antibodies bind and have a wonderful structure function immunology paper demonstrating the deep immunology at the antibody binding level of how myasthenia gravis occurs, all the way down to the antibody on the receptor. And they find key regions of the receptor that are important. They see how the loop moves to open up the pore in the channel. They see how things can stick into the channel to block it. So even if you have drug binding, the antibody doesn't let the ions go through. It's a pretty cool tour de force of getting this all done. Other cool deep insights besides, wow, all these antibodies do things, and here's some structure and what that really functions. Because these are all monoclonal antibodies, right? They then map all of the different antibodies on top of each other and see their regions of commonality, which is probably where you have problems with polyclonal antibodies as well. But if you could take a mini protein structure of those regions that are common and dope that into people, you could sync the antibodies, right? And then get them out of circulation from being able to bind to the protein. So pretty dang cool.
So I'm looking at the graphic abstract of the paper. So I see the acetylcholine receptor. So it's important for muscle contraction, right?
It's required for muscle contraction, right? And so when acetylcholine binds, and ion channels go through, and then I think it's calcium, if I remember, calcium influx, and then muscle.
Yeah, perfect. So here I see that it has these little structures it's like a conical structure that goes through the tip of which goes through the double.
Yep, standard transmembrane ion channel design.
And so there's, in this case, I see this different antibodies. So
they're showing where different antibodies would bind and how they then mess up different things, right? So some antibodies will bind the binding site, or cross-link receptors together, activate complements. Some block the ion permeation. So if you look at like the graphical abstract, the thing right in the middle of the channel, an antibody bound there, well then don't matter if acetylcholine binds, the receptor can't get in, right?
Right. So, but then I see two different things. On the one hand, is the effect of uh, preventing the action of the channel. So that I guess that would
Yep.
impair
And
muscle
most of the
contraction.
antibodies do that a little bit, even if that
Mm-hmm.
they have other functions, they'll stop acetylcholine from binding or block the pore. The other one
Mm-hmm.
is it causes internalization of the receptor
Right. And that also reduces the
or
signaling.
complement, which then kills the cell.
Exactly. So that's the point. Why. So there's different, and they can different antibodies will have a different tendency to
Yes.
does it
And so they picked
to
antibodies
mediate these
with
things.
known different properties of how they work and then mapped what they did.
Okay. And I would expect them. So when it comes to, I guess it's mostly focused on the receptor itself, but when it comes to the type of antibodies
They
that
looked at a
are
couple
involved.
IG1 and IG3, but they looked at IgG only.
Okay. All right. And they're all equally capable of, for example, activating complement
No.
or
So that's like, so
no,
the example
definitely
like
not.
antibody 1B in their list activates complement. Antibody 2 activates complement. And those tend to be on these winged structures.
Right. Because the activation of the capacity to activate complement will depend both on the type of FC, as the
Right.
constant the receptor or the type, and also whether I guess if the receptor gets internalized, then there's not going to activate complement. Is being
Right. But
if
they
the
found that
whole thing
mostly
is being internalized.
cross-linking receptors lead to the complement activation. The antibodies known to cross-link receptors seem to activate complement.
Okay.
So yeah, it's really but like getting a structure of this. I didn't realize we did have one, and then being able to have the antibody bind to it is pretty
Yeah.
cool.
Yeah, very important in order to understand, no, how where is this disease coming from? How is it establishing? I guess if you have all this B cell response, you probably also have a T cell response that is also reacting to some of these epitopes in this maybe in the same domains as the antibodies detect. Very interesting. And well, if we're gonna be talking about autoimmunity or in my case, I guess more like allergy, but on this misaligned immune responses. I have a really nice story from Science Immunology. First author Daniel Weissman, from corresponding authors Anna Einstein and Andrew Wang from Yale School of Medicine. And the was published in April 4th, and it's called Skin Damage Signals Mediate Allergic Sensitization to Spatially Unlinked Antigen. And I think this is really, I mean, I think it's a very interesting publication, very interesting paper, because it really helps us understand the origin of allergic sensitization. And it really feels a bit unexpected, the results. And I wonder, what the implications are to therapy or to understanding when people get allergic responses. Where are these stimuli coming from that initiate these responses? So allergens, people get allergy against all kind of things. Here they're focusing on, the mucosal allergies, uh, things that, food allergies, allergies of things that you breathe. And I think it's been quite accepted by now that you can initiate a gut allergy or like a mucosal allergy after being immunized in the skin. So there has already been research indicating that if you are exposing damaged skin to an allergen, that can result on uh, mucosal immunity. That so, for example, if you got an ova, if you're scratching ova, there's this models of skin damage, and then if you expose this skin to food allergens, then you can promote this allergy against this food.
That's how my wife got a zucchini allergy. We gardened zucchini and she got her skin scratched gardening the zucchini with the little burrs on the plant, and now she can't eat zucchini, which was her favorite vegetable.
That's very unfortunate. But yes, I think this is kind of the idea that this paper is going after. I mean, that already is kind of insane, right? It's like it also feels like there's nothing safe anymore. You can just get allergies from, from damaging your skin. But that aside, I want to make it even worse for you because in this publication, they show that you don't actually need to have the antigen present at the same location as the skin damage in order to induce allergy. So nothing safe, nothing sacred. You can get allergy in multiple ways that you weren't expecting. So what did they do? They have this model of what they call remote priming. Basically, they have this mice and they proceed to administer different forms of skin injury. The most common is called tape stripping, in which they basically put a tape on the skin and they just pull very quickly, and then that's how you get all the surface layer of the skin out. They also do some other things. They damage with acetone. They do a punch biopsy. They have a UV radiation. So quite several ways of getting the skin irritated. And they administer OVA peptide, our favorite model antigen for mouse allergy and other immune responses, but not on the skin as you would expect, and it has already been tested, but via oral gavage. And they go through certain lengths to show that it is really the exposure of the antigen in the gut, and not any accidental exposure to the wound site or to the skin site. And they show that by doing this, they can actually find antibody titers and anaphylactic responses after administering OVA through, I think, IV. Yeah. So they see OVA-specific IgG1, IgE, and they show basically systemic anaphylaxis when they get challenged with OVA later. So it shows that they can have the OVA in the gut, the damage in the skin, and still get the mice allergic to OVA. And they coined this effect as a remote priming. And when they look into the mechanisms behind this connection, they basically try to identify what is the message that's coming from the skin to the gut, and how is it that you're getting primed this way, or the mice are getting primed this way. And they do cytokine profiling in the plasma after the skin injury. They do a lot of measurements of the timing, kinetics. So I'm not going to go over that, but basically they do some thorough investigation of cytokines, and they find a bunch that are consistently elevated across different of the skin injury models that they look into. And I'm going to here mention particularly IL-33, IL-1 beta, IL-18, TSLP, and then they measure other cytokines that are also elevated, but they show that these are the ones, IL-33, IL-1 beta, IL-18, TSLP, that are really correlated with the production of IgE and IgG1 and this anaphylactic response. Basically, they double down on this IL-33, TSLP, and another cytokine called IL-15, which are known kind of collectively as these alarmins that seem to be mostly the most important ones for mediating this remote priming. And they even, if they show that no, there's a lot of, I'm leaving out because they do a lot of experience showing the timing. I think very, very important, the timing. They do mouse knockout models, and they show that if they focus in particular on IL-33, which I think is the prototypical alarmin cytokine, and if they co-administer IL-33 with a distal OVA immunization, they can, this is enough to prime this allergic reaction. So basically, they show that by using IL-33, they can induce a high affinity IgE production, and they even work for other antigens. For example, they give mice a peanut allergy, for example, using this mechanism. This mechanism, and what is interesting is that they also show that this only works for previously unknown antigens. So if the mice had already been exposed to ova in a tolerogenic way before, then this is not enough to elicit an allergic response upon IL-33 and antigen co-administration. So basically, who are the critical mediators here are, and surprisingly, ILC2s, which we're going to be talking about today, later today. So ILC2s, which are known for high expressing, high amounts of these ST2, the IL-33 receptor, are critical for this IgE switching and the follicular helper generation necessary for this immune response. And then they are producing IL-4, IL-13, and this is kind of how they end up priming the immune response towards an allergic response. So basically what they show is that you can have damage in the skin, which will release alarmins, particularly IL-33. So they did most of the mechanistic studies with IL-33, and it is this IL-33 that will activate ILC2s, which in time will produce IL-4, IL-13, and all this kind of succession of events is important for linking this epithelial damage to a systemic sensitization to an antigen that is being exposed in the gut, for example. So I thought it's just crazy. There's no safe place here.
So no matter what you do, you're going to get an allergy unless what if you eat everything though?
Well, I think that's the thing, right? If the mice had already been exposed to the allergen before without skin damage, then that wouldn't work. So if they give the ova or gavage the ova before, or like they gave it ova in the water before, and then they tried doing this, then that was not enough to elicit to establish the allergic sensitization. So yes, you should eat everything whilst you are in a very non-inflammatory, very controlled environment, and then hopefully that will protect you against future potentially problematic exposures.
There you go.
I mean, it's just insane. I think the data is solid, but it just feels like so hard to believe that it's just, you can have this weird connection.
Wild. That is super wild. All right. Well, I don't really have a good segue here, but I do have a tour de force of signaling for you, which is Epicalus induces intestinal T helper cell 17 cells by promoting E3 ubiquitin ligase TRIM21 mediated E3 ubiquitin ligase FBXW7 degradation. We're going to get
What a mouthful.
Yeah. It's in immunity. Prepare for your second mouthful, where I will coming out in June 10th, 2025.
Wow, you are living in the future, Jason.
I'm moving fast. All right. First authors are Ying Shen, Zheyu Ma, Hanling Wang, Yinghu Chen. Last author, Zijian Gai. Hopefully I did not commit any terrible butchery of that. All right. This paper, I'm going to start by saying there is a known effect that FBXW7, which is a ubiquitin ligase, is important in T cell development. So one of the problems they had with studying things is that if you just knock it out, you don't get T cell development. So they created a distal LCK Cree mouse where Cree expression is delayed after positive selection. And they crossed that to an XW7 flox mouse. So this only takes effect after you have positive selection. And they found that knockout increases Th17 cells, non-pathogenic Th17 signals, and all of that. And then they tried to figure out how. For an entire paper with a lot of knockout mice and luciferase reporter assays and all this stuff. So I'm just going to explain the graphical abstract because it's going to take a while. All right. So, and then I'm going to talk about the bacteria. But there's a bacteria that induces this. But what happens is, so the other little bit, sorry, I'm going to go back one step back. There, segmented filamentous bacteria are a big thing in mice that are thought to be protective by being present against pathogens like Citrobacter. But in human, we don't have very good understanding of if they're there or not. So they talk about this here. I actually think this is the weak point of the paper. But they do find a bacteria that they culture, and it's this A. efficalis. It's non-pathogenic, but what does it do? So they show, and I'm going out of order here because it's better to tell this in the signaling order than the other one. They show that microvesicles, as well as proteins from this, not other things from this, from the supernatant of cultures, but proteins, but they don't know which ones, do something. So either the vesicle, which is endocytosed, or the protein, which is uptaken by CDC42, as a vesicle, right, through endocytosis, but either outer membrane vesicles or endocytosed protein through CDC42. That inhibits TRIM1 autoubiquination. TRIM1 is ubiquinase, it ubiquinates itself, and then goes away through an interaction with UBE2W. So this protein, whatever happens, this bacteria causes UBE, breaks the UBE2W TRIM1 complex, and thus inhibits TRIM1 autoubiquination. What does TRIM1 do? TRIM1 degrades FBXW7. Loss of, I'm just going to say W7, right? That is what they initially studied in the knockout mouse. That increases T helper cell. Okay, so getting rid of this bacteria coming in stops TRIM1 from eating itself. TRIM1 can then go degrade and drop the levels of FBWX. So that's how the bacteria induces Th17 profiles. Well, how the hell does it do that? Well, W7, that ubiquinase, that ubiquinates Jun and degrades Jun B. So now if you have less of it, you have more Jun B. Jun B goes and transcriptionally activate is in the promoter, binds the promoter of the aryl hydrocarbon receptor, which is also a transcription factor. So aryl hydrocarbon receptor is transcribed. That goes and binds the promoter of ROR gamma C. And that makes you have more Th17 helper cells. They map out the whole thing, the whole kit and caboodle.
Yeah, I'm looking at the abstract myself, and it's like it's a lot of intermediate steps.
The graphical abstract is where you got to stay.
Yeah,
it's a lot of steps.
All right, so what is the? Can you remind me? You said, and what is special about this bacteria again?
This is a non-pathogenic segmented filamentous bacteria, which are those bacteria are very rarely found in people. We know we have them, but like, are we colonized with them is in dispute. They say they can find some isolates, but it's like they found a little bit kind of somewhere. But they basically do all this in mice and try to drag claim to humans. The reason it's in immunity and not like cell or in nature is I don't think they have the human connection solid enough. But they are showing that commensal mice bacteria, at the very least, which they think also are in people, drive this reaction to drive, homeostatic Th17 cells.
Right. And so in this case, so we want homeostatic because...
You want homeostatic Th17 cells because they help protect the host from damage during, pathogenic bacteria. They can go wild and cause autoimmunity too, for sure, but they have
Right.
a really important like homeostatic effect, right? And so then they map out the whole thing, how this bacteria is inducing it, which is through, three ubiquinases of regulation combined with two transcription factors.
That's pretty
Or I guess ROR
complex.
gamma C is also transcription factor, isn't it?
Yeah.
So three.
Yes.
So three.
Yeah. Okay. Okay. I guess, yeah, I guess you need, so these Th17 cells, you need them in order to have the homeostasis in the gut. You don't want too much though, because as you said, that would also, there's a balance to this whole situation. Is there anything about having too much IL-17 or Th17 response in the
Not in
gut?
this. It this only seems to work in non-pathogenic conditions to begin with.
Right. But are they exposing the mice to pathogenic bacteria to see how,
Not in this
like,
study. No, they spent all
okay.
the time mapping this out. It's already known if you get rid of Th17 cells and then go give mice Citrobacter, they die.
Yeah, okay.
It's
That was around what, so
yeah,
they, but
very,
they
very,
didn't
very
test it
no,
here.
known long ago in a galaxy far away.
Okay, because I guess also that's also important because one thing is that if you have the good bacteria, you can say, well, they're just competing with the bad bacteria for space or they're competing
Yeah,
for nutrients. But this is a different mechanism that includes the response of the host specifically to this bacteria and the activation of immune pathways in the host that protect against the pathogenic bacteria. So it's not the same.
exactly. This is different. This is the host. This is the bacteria altering the host to affect other bacteria.
All right. Well, that's pretty interesting. There's so many layers of regulation here. It's just dizzying. Okay, so why don't we finish our roundup today? And I have another really cool story. And I think there's already a few papers that discuss about your sex differences and also like neuroimmunity. It's just, this is all the rage. And I just keep finding these stories and they just keep coming and they're so interesting. So let me share with you this paper that is called Meningeal Regulatory T cells inhibit nociception in female mice. This was published in Science. First author Elora Midavine, I'm not sure how you pronounce it, from corresponding authors Alan Basbaum and Sakim Kashem at UCSF. And it's just, well, already the title says a lot. And I don't want to make, big, this is all in mice, right? It's not, but, I think it's pretty established that there are differences in pain perception between females and males. I think this is, has been shown in humans and there's, I'm not sure what the mouse situation was, but I guess this is also something that people have observed in mice. And it's interesting that regulatory T cells coming to the mix because yeah, their transcription factor that kind of defines CD4 regulatory T cells is this FOXP3, which happens to be X-linked. And it's interesting because that makes it a little bit harder to something to study against Tregs because then you can say, well, what about the two copies that technically cells have? I mean, one of them is supposed to be inactivated during the regular X chromosome inactivation, but I guess it's hard sometimes to differentiate the effect of like genetics and the effect of hormonal differences between males and females. And I think by now we know that regulatory T cells interact with, the nervous system. We know that they are present in the brain. So we also discussed this in the Roundup before. They are in the meninges. They're important for recovery after a traumatic head injury. So we've also had some of our guests discussing that. So it's sometimes it really makes me think, is there something at T-Rex are not doing? Because they're everywhere and they're doing all these odd jobs sometimes. And in this case, this is an example of how T-Rex are somehow adapting to their location, and they are doing things that are not necessarily the first thing you would think about when when you think of the traditional role of a regulatory T cell, which is to inhibit inflammation or inflammatory T cells. And I think there's more and more evidence that they do also other things. So let's talk about this paper. Basically, we know that Tregs are important. They are present in the meninges. They are also present in this in the dorsal root ganglia of so it's different parts of the nervous system, and it has been seen that there are Tregs present. And so the question is, what are they doing there? What is their function in this particular spot? And they in this they show they look to in this paper they start describing this distribution and showing that there's this enriched population of Tregs. And they show that if they can selectively deplete this meningeal Tregs through intrathecal injection. That's in the brains. Is that in the brainstem? So within kind of the CNS compartment, and they have this model of what is known as the kind of the diphtheria Treg, the FOXP3 TR mice. And basically, you have a diphtheria toxin receptor that it's associated with, in this case, FOXP3 expression. And then if you have that, and if you inject the mice, systemically with diphtheria toxin, then you get rid of all the Tregs. But if you inject a pegylated version of the diphtheria toxin inside of this central nervous system, you can reduce the Treg populations inside the meninges, this meningeal population, very specifically, without really affecting systemic Tregs. So you can really separate the effect of one of the total Treg population versus the ones that are living in this particular niches. And what's interesting is that when they do this in these mice, they show that this depletion caused a increased mechanical hypersensitivity, but only in female mice. And this had no effect on other kind of reception properties of unwanted kind of stimuli like heat or cold or the motor function. So it's only in this very specific mechanical pain sensitivity is affected only in female mice when you deplete this very specific population of meningeal Tregs. And when they look at it, so they do that, and they do some models of in which they generate nerve injury to generate pain. And again, they really show that in this case, the depletion of the meningeal Tregs increases hypersensitivity in females, so that it looks like these Tregs are necessary to protect the females from the pain caused by this spared nerve injury model that they do. They show that if they inject low dose IL-2 in the CNS, this reduced the pain after this spared nerve injury model, SNI, but only females. So the increase of Treg populations through IL-2 low dose, you see an increase of Tregs, and this correlates with a reduced pain in females. And so basically they conclude that Tregs can modulate this neuropathic pain. And this has nothing to do with the more traditional function of Tregs of mediating tissue repair or inhibiting other T cells. This is really something else. And they also show that this is because of the hormonal composition of the mice. They have mice that are expressing one or two X chromosomes, and they're female. So they have mice in which they have two X chromosomes that they're still male because they're still expressing the part of the Y chromosome that makes them male in a different chromosomes. It's a very specific mouse model, and this really shows that is the hormones, the female hormones that are underlying this pain suppressive roles of meningeal Tregs. And if they are, remove the ovaries or they have this estrogen blockade, they again abolish this effect of the Treg. So it really has to do with a hormonal dependent mechanism. And what they zeroed down is that Tregs, this particular population of Tregs, can express a opioid peptide called enkephalin, and which is a opioid peptide that was a peptide that can bind to the opioid receptors. And this is how they are kind of mediating their function. And so they see PENK, the proto hormone production in the Tregs. They see it in Tregs specifically. They show that if they treat with IL-2, they can increase enkephalin in the CSF, in the cerebral spinal fluid. And they really show that if they deplete PENK producing Tregs in females, this reduce the pain thresholds of these mice. And so again, and this has nothing to do with the traditional suppressing function of Tregs. It's just the capacity of suppressing the pain perception in the CNS. And they just show that the targets of these enkephalin produced by the Tregs is this delta opioid receptors that are expressing a specific subset of neurons that are mediating the pain, this mechanical pain. And basically that's how they make the whole connection between the Tregs in this particular site, production of these opioid peptides, these peptides that bind to the opioid receptors, and the capacity of influencing these sensory neurons that are mediating and this pain perception. So yeah, I guess I don't know why would females have that? I don't know, just to be better at what is the advantage of having this pain, very specific pain since resistance. It's very interesting to kind of hypothesize what is the meaning of this.
Childbirth.
Yeah, I guess that is the most kind of, but it really is. Yeah, I guess because then they would never do it again. I don't know.
Exactly.
Probably.
Good question. All right. Well, we're going to be speaking to Dr. Henrique Veiga-Fernandes, the Champalimaud Foundation, in just a moment. Before we get to that, we'd like to remind listeners to register for IUIS 2025. The International Congress of Immunology is happening August 17th to 22nd in Vienna. Check out the program and see the confirmed keynote speakers at iuis2025.org.
Now for the second part of our show for our interview, we're really happy to welcome to the podcast Dr. Henrique Veiga-Fernandes. He is a full investigator and group leader at the Champalimaud Foundation in Portugal. And the Veiga-Fernandes lab studies the role of the crosstalk between neurons and the immune system, has really some really cool, unexpected research on ILCs and what a really, what would say, pragmatic group of cells they are. It's really interesting. And I guess we're going to be talking about some of his recent research. Thank you so much for joining the Immunology Podcast today.
Oh, my pleasure. It's really a pleasure to be here with you today. So thank you for having me.
Thank you for being here. So, I keep thinking about your science paper that just came out with glucagon neuronal ILC2 interactions and how you have a brain-gut axis controlling, essentially glucose metabolism. So could you maybe, for those who haven't read that paper, give us a quick summary of that relatively crazy story?
Yeah, with pleasure. So the old message at the end of the day, so what we did find was that once you go into fasting, so the consequence of that is our blood glucose level or sugar level go down, obviously, because we don't have exogenous sources of glucose. That triggers an endocrine response that is characterized by the production of an hormone in the pancreas. That hormone is called glucagon. And what glucagon does is to trigger metabolic processes that will use fats and proteins to produce endogenous glucose. And so we get a stable level of blood glucose. And that allows us to, still think or run appropriately even if we don't have access to food. So you can imagine that's tremendously important from evolutionary perspective. So what was very unexpected was that we found that in addition to the normal neuroregulation in the pancreas that happens with glucagon, when we had mice that were fasted, that would trigger a brain-gut axis that would increase the activity of sympathetic neurons in the proximal part of the gut. And when you activate sympathetic neurons, that leads to the release of a neurotransmitter called norepinephrine. And norepinephrine would now provide an instruction to innate lymphoid cells type 2 or ILC2s that are in the intestine. And that instruction is go to the pancreas. And they go to the pancreas, they migrate to the pancreas, where via the production of type 2 cytokines, now they induce the production of glucagon from alpha cells. So these are important endocrine cells present in the islets of Langerhans together with beta cells. And this glucagon, once it's produced, now will go through the bloodstream and act in the liver where it can trigger gluconeogenesis. So in other words, the hepatocytes will now use the lipids and proteins to produce a glucose. So we found here really a neurocommunication in the intestine that leads to an inter-organ communication from the intestine to the pancreas. In the pancreas, there's the production of glucagon that now triggers gluconeogenesis in the liver.
Definitely unexpected. Can I maybe take a step back? One quick question. You mentioned type 2 cytokines. Which cytokines would fall in that category?
Yeah, so in the case of ILC2s, right, they produce interleukin 5, 13. They also produce large amounts of amphiregulin, and they can produce also enkephalin peptides that are very, very important in other contexts, notably in the adipose tissue. But in this specific setting, the most important cytokines are IL-5 and IL-13. And why? Because alpha cells in the pancreas express interleukin 5 receptor and 13 receptor. And so if you provide these cytokines, that induces increased secretion of glucagon. Actually, a lot of cell types in the body express receptors for these type 2 cytokines, IL-13 and IL-5, and these endocrine cells are not an exception, so they do produce them.
So for the listeners, I covered this paper on episode 97, by the way. But now this is 103. So, that's been a few months. My question, I guess, is why do you think the body evolved this way? It seems remarkably indirect, I guess, to have this many layers between cause and effect. And yet we obviously evolved this way. So there has to be some type of fine-tuning reason for it. But it's crazy.
Can I just add, maybe there is, from my biochemistry classes, right? There is a very well-established mechanism by which, independently of the immune system, the absence of glucose can be sensed and the signaling can go to the liver to undergo this procedure. So maybe would you like to say what was our understanding so far, and maybe why do we think we need this extra layer of complexity added by these immune cells?
Yes, Brenda, you're very right. So there's a completely immune cell independent mechanism of glucagon regulation. So when glucose levels go down, the same sympathetic tone increases directly in the pancreas, and norepinephrine can directly regulate glucagon production. And actually, when we compared animals that had no innate lymphoid cells type 2 and compared glucagon levels, it's not like the glucagon disappears. There's still some glucagon, but much lower than when you put back ILCs. So which means while there is a neuroendocrine loop that regulates glucagon production directly, now there's an extra layer, as you were referring to, that requires immune cells. Now the question is, why did we evolve like that? And I don't have a straight answer for you because that's a difficult question. I can speculate reasons why that could potentially happen. So we found actually on these alpha cells that produce glucagon, these very high levels of type 2 cytokine receptors. And these cytokines, in addition to being produced very high, being produced by these cells and expressed at very high levels in endocrine cells, they respond to type 2 cytokines that are produced obviously by innate lymphoid cells type 2, but also in other contexts. One of those contexts actually is in the context of worm infections. So, and worm infections, in particular nematodes or helminths, actually have been evolutionary partners of mammals. So if you look into the wild and you ask how many animals are infected, you're going to have a very, very high percentage. Virtually every single animal will be infected with helminths at some point. Just think about, and for our audience, if they have pets at home, dogs or cats, they go to the vet, and one of the treatments they get on an annual basis is actually a treatment against helminth parasites. Why? Because they are endemic everywhere, and you get them, right? And this, what it triggers is a constant higher level of type 2 cytokines. So one possibility that we could think here from an evolutionary perspective is that associating type 2 responses to increase glucagon would be mutual benefit for both the host that needs to increase energy levels because you've got a parasite consuming a lot of your energy, but also for the parasite itself because it has a host that survives much better for a prolonged period of time. So that is a good, an interesting hypothesis that we didn't test. Obviously, it's a difficult one, but it's an interpretation of what might be happening. The second one is that this might have been a process that has been triggered during evolution to really maximize the amount of glucagon and therefore to maximize the production of endogenous glucose production in a time-dependent manner. So the direct mechanism that you mentioned, Brenda, where you've got sympathetic neurons directly regulating glucagon production, this happens very fast. So you have this happening already at a couple of hours after fasting. While what might be happening is that for prolonged fasting, now the system needs an added layer that increases even more glucagon so that you can keep running, you can keep surviving for longer during fasting, even if you don't have access to food, right? So if you take rodents, if this would last more than, 16 hours, more than an overnight period, 24 hours, now you would have this extra layer that would still provide you the energy for you to run from predators or the energy that your brain would use to think a little bit and try and identify the strategy to forage for food and extracellular or exogenous energy, right? So these are obviously speculations, but I think those would potentially make sense in the context of our findings.
So I am always baffled by the kind of unusual tasks that cells like ILC, particularly ILCs, what is up with them? Always, they really are one of the examples of the idea that the immune system is so much more than just keeping infections at bay. They're always intervening in so many processes that we wouldn't expect them to. You also have other research in ILC3s, and you also have some of your earlier research also looking at ILC2 in and neuronal regulation. So maybe would you like to share with the listeners a little bit of a primer of ILCs doing unexpected things, just for us to understand, like, really what is that we are up against when you're researching these cells.
Yeah, sure. You're very right. So, when these cells were officially discovered, and they were now discovered, I would say easily, about 15 years ago. The whole idea was, in essence, to study what would be the role of these cells in immunity. And this was obvious that they probably would do something related with immunity because they were producing the same type of set or the same set of cytokines as T helper cells, right? In the specific case of ILC2s. And actually turned out that they are absolutely critical, especially in the context of helminth infection. So if you want to go well with infection by helminths, you need ILCs, and specifically in this case ILC2s, because they are the first producers of large amounts of type 2 cytokines. No surprises there. Surprises started to emerge when we actually, as you mentioned, figured out that these cells are also extremely important for physiological processes of the host, even in the complete absence of infection. And why do they do it? Well, there is a big difference between innate lymphoid cells and adaptive lymphocytes. And probably the biggest difference is that they are present and they are constituents of organs and tissues. So they are always present in non-lymphoid tissues, the vast majority of them. Especially for ILC2s in the context of mice, for example, they are in the vast majority of organs where they produce, in a almost innate fashion, they produce these cytokines, type 2 cytokines that we already mentioned. So IL-5, IL-13, amphiregulin, and metaencaphalin peptides. And it turns out that in the body, many of those cells express receptors for these cytokines. And the functions of the signals through IL-5 and IL-13 receptor, then it will depend very much on the target cell. So it's a very much, I would put it, cellular context-dependent manner. So depending on the receiving cell, now the output will be very different, right? For example, give you an example for ILC2s. The one we already described with endocrine cells, especially alpha cells, who produce significant amounts of glucagon. But for example, if an adipocyte sees IL-5 and IL-13, now we will turn on what we call an energy expenditure program. So it will start using lipids to increase body temperature, right? So this layer of functions of innate lymphoid cells actually very much falls within a concept that I think is now growing momentum and interest from the wide community, which is the fact that the immune system, in addition to protect us from infection, in other words, in addition to their roles in immunity, that can be infection, cancer, you name it, actually, they are very important regulators of body physiology. By regulating how other cells interact between themselves, how other cells produce functions that contribute to the homeostasis of a given organ, or even to how they regulate, for example, metabolism that has an impact in every single cell in the body. So I gave you the example of glucagon with the glucose. We also showed in the past the importance of these cells in the context of lipid metabolism. So they really play here very important roles. We now also have ideas, very clear ideas of how these cells, for example, contribute to brain development and to contribute how cell or neurons establish synapses in the developing brain. So it's, I'm pretty sure there are many other aspects and many other processes that we're still, they're still out there and we have no idea. They're there for us to discover what they might be.
So do you think ILCs then co-evolved, or at least ILC2s, with parasites in multi-organ systems? And do we know if evolutionarily they seem to have merged at similar times? Has anyone looked into that?
So actually there's pioneering work from Max Cooper, where they actually saw that lampreys already have some sort of ILC-like type of cells. So these are really very likely very ancient cells, right? I mean, fish also have ILC-like type of cells. So one possibility is that you could think about it, that they evolved with infections that would require type 2 responses. But you could think about these also very differently. Perhaps what I'm going to say is a little bit provocative, but you could also envisage that their primordial function was not really immunity, but rather regulation of physiology in simpler organisms. And then their functions might have been co-opted for immunity functions, right? Um, so, because if you think about it and you start looking at what cell types express type 2 receptors, you will find a huge variety of cell types expressing these receptors. So now, especially with these omics, that we have databases of anything and everything you can think of and ask what kind of genes are expressed by cell A or cell B, you will see that they are distributed in a large range of cell types. So maybe, they were co-opted by immunity. I think it would make a lot of sense because we're seeing things like these regulation of glucose metabolism is something essential to life, right? I mean, it's really critical. Or maybe the two things happen throughout evolution almost in parallel, right? Where you would have these processes occurring almost in parallel, coevolution with type 2 responses, classical immunity functions, but also regulation of physiological processes.
When you think about it, you mentioned 15 years or so since we've been aware of these cells. And that's not a really long time when you really consider it. So it's really kind of very interesting to think how late they have been discovered. And I guess that has to do with what you mentioned about ILCs being mostly found in tissues and how, we've been improving our techniques for recovering, because also these cells are not very abundant, right? So what percentage, how abundant? So to give an idea to our listeners that might not be, too familiar, how difficult is it to really study these cells? I would say in mice, but also, from humans.
Yeah. No, absolutely. So nowadays, the ILC family, in addition to uh, uh, helper ILC 1, 2s, and 3s, they also include a family that was discovered much earlier. NK cells are now also considered part of the ILC family. And NK cells are considerably more abundant than any of the other subsets. Concerning the other subsets, the first one that was identified, actually, there was pioneering work essentially from brain amoebias working at the time in the Netherlands. They are very abundant. There's a type of innate lymphoid cells, we call them LTI, and that means lymphoid tissue inducer cell. They are very abundant during embryonic life. They are a subset of cells that fall within the large family of innate lymphoid cells type 3. And the role of these cells is to induce the formation of secondary lymphoid organs. So without these cells, you don't get lymph nodes, you don't get Peyer's patches, and you get an organization of the spleen architecture that is deficient, right? So they are very important and very abundant at this stage. Once you get in adult, that's where the difficulties start. And why? Essentially because they are in tissues. So there was a technical hurdle there to get sufficient number of cells because they are quite produced. But they were also present in lymph nodes, for example, but their number in frequency was very, very low. So, in immunology at the time, we were mostly detecting these cells by flow cytometry, and the percentage was that one very close to what you consider as a background, the hot dot that appears in your screen, that is background. So nobody really paid much attention to that. Until some groups started actually to look at these cells in animals, especially in rag knockouts. Now you don't have adaptive lymphocytes, and those percentages were a little bit higher. And then the real change and the real breakthrough was when people made report mice for cytokines, and they realized that there were cells expressing type 2 cytokines or IL-17 and IL-22 that were not classical lymphocytes, and they were essentially present in tissue. So now this could not be a background anymore, right? Because you know, not only you were seeing the cells, but you could also allocate the production of these cytokines to cells that were, you know, lymphoid in appearance, in shape, format. They expressed many molecules similar to adaptive lymphocytes, but they didn't express the classical BCR or TCR molecules that are specific to B cells and T cells, respectively, right? That's when they were discovered. And since then, when you think about it, 15 years, we now know a lot about these cells, you know, all their transcription signature, the lineages, their transcriptional requirements, their functions. And this was heavily fueled by what we knew already from T helper cells, because actually the vast majority of the transcription factors that control the acquisition of cytokines by the subsets of ILCs are very similar to the ones that control the expression of these types of cytokines in adaptive T cells.
You really got me thinking about that, no? The idea that adaptive immunity is just repurposed ILCs in a way. That kind of hurts my brain to think it about that way, because of course we think about adaptive immunity and T cells, and now we've been, you know, swimming in their knowledge for so long that they've been there forever, and ILCs feels like a newer concept, but then it completely makes sense that evolutionarily they came first, and then adaptive immunity can be an addition to this pre-existing or something like ILCs, right? But the idea that adaptive or like these receptors, antigen receptors or proteins are a much later invention, that cells that can make cytokines in tissues.
It could be. They are, at least when we look in terms of models, they probably are as ancient as some initial lymphocytes with some degree of rearrangements, right? Maybe not the classical or the T cells or B cells that we see in mammals, but something like that, right? Where you would have already cells with features that highly resemble ILCs. Of course, there's a fundamental difference between them. And I think this is also important for the audience that is not so familiarized with ILCs. So how do they get activated? So we all very likely know how B cells or T cells are activated, and you get an adaptive response where a given T cell, for example, with a specific or with a discrete T cell receptor or TCR will recognize a peptide. And if it turns out that peptide, for example, is a peptide that is present in one type of bacteria, now you're going to have this T cell that recognizes the peptide presented in the context of MHC class 1 or class 2, and you've got clonal expansion of those cells. So you're going to have millions of cells bearing the same TCR or BCR responding specifically by killing infected cells, for example, by producing antibodies to that specific invader. In this case, it could be a bacteria or a virus, it doesn't really matter. Now, innate lymphoid cells are very different, right? Although they respond to very specific signals that really rely essentially on what I would call cytokine signals that are produced by other cells in the body. And those include mesenchymal cells, epithelial cells, that produce a set of cytokines that we call or coin alarmins, that give an instruction, oh, there's something wrong here in the tissue potentially. For example, in the case of ILC2s, they respond very, very strongly, essentially, to two main alarmins, IL-33 and also IL-25. And these are alarmins that are abundantly expressed when there is some sort of damage induced in a barrier tissue, right? So these are the differences. And the consequence of this is that the timing of response and production of cytokine now will also be very different between innate lymphoid cells, so in between ILCs and lymphocytes. So while ILCs can respond in a matter of hours, if not minutes, now for having a primary response from T helper cells, that will take you a number of days, up to 4 to 5 days, to get a tangible number of T helper cells that now can fight infection, for example, right? So you have an initial phase already of protection that is essentially controlled by these innate lymphoid cells. But they actually, at the end of the day, the functions are different, so they are not redundant at all in terms of the immune response, but they act, or their effect occurs at different phases of the response, with ILCs as early responders and T helper cells as late responders. When there's no infection, ILCs, what they're doing is still producing these cytokines, but they will be regulating many physiological processes, as we've discussed before.
So I know we could keep going down this further, but I wanted to kind of segue, because you have an interesting thing we haven't had a guest on the show before which is you've been in GQ as Man of the Year.
So maybe you need to clarify what GQ is. I don't think everyone knows.
Oh, Jason, you probably know what GQ is better than I do.
It's a fashion and popular culture magazine. And you were GQ Portugal's Men of the Year Award in Science. So I have to ask, what was that like? Did you get to have a photo shoot? And tell me more.
I don't know how you get that, but so yeah, this was a period. I don't know if they are still doing it, but GQ Portugal was doing once a year. They were choosing people from different areas in the country. It could be science, it could be music. There were several categories, and they would select one individual. I mean, this is essentially, at the time, was essentially male, so only men for some reason. I think this is a magazine for men, if I'm not mistaken, or not for men, but related to men activities, I guess. And here you go. So it was fun, because there was a photo shoot. Suddenly I was like, oh, what is this? Like a model. And then there was a party with all these, after party photo sessions and everything. So it was a bit like, a fish out of the aquarium, if you see what I mean. Very different environment from what you were used to when you are a scientist. But it was big fun. It was quite an experience. And at the end of the day, I think these events are also quite important, because you bring science and the importance of science to a community that normally is not exposed to science whatsoever. And getting these kind of magazines or media interested in science is quite spectacular. I find it was not just the photos. There was a small interview, so you get to talk a little bit about science and the importance of science. And I think this is an interesting outreaching activity, not to just see scientists as the weirdos on lab coats that are, hours and hours in front of a microscope that never get out of the lab. We're just human beings like anybody else. So it was nice. It was fun.
Indeed, human beings like everyone else. I like that. That's nice. Sounds like a fun thing to have experienced, and it's good, that you are there, getting yourself out there as a role model for people reading pop culture magazines. I think it's always good. I don't know what you think, Jason. You might be part of the target audience of such a magazine.
I do not read GQ.
Okay.
I mean,
well,
not very much. I've read it a couple of times, sure, but
when I was waiting at the doctor's office for my appointment.
Something like that. Like, I mean, you've seen me dress at conferences. I don't, I wear Jedi robes.
So you're not like everyone.
Well, I'm not a reader either. I'm not a GQ reader either. It was, one of those episodes, that, yeah.
Yeah, unfortunately, we couldn't get a hold of the actual article anymore. So the link is not working. So we have not been what's the word, delighted with seeing your photo shoot, but I'm sure it went really well. I'm sure it went really well. All right, I think we are reaching the end of our conversation. We like to ask our listeners like a little question to get them to know them better, give them opportunity to say something or share something with our audience. I have a question for you. You let me know if you are, if you're prepared to answer. If not, I can also bring another question because we did not warn you in advance. But I think something tells me that you might have an answer to this question I'm going to say. So, a popular thing to ask for us is if you had, a piece of advice that somebody has ever given to you that, could be professional or not, that you would like to share with us today, what would it be?
Yeah, I mean, I think every single scientist has people that, a number of people that for one reason A or B were major influences in their career, right? Everybody has got that. Um, but I probably would highlight something that, in a discussion I had at the time with my PhD supervisor, we were discussing about the issue that at the time was already happening. I did my PhD in France. And what was happening was that especially young people were not motivated to become scientists anymore for multiple reasons that are still matter of debate today. Getting a career, stable career, work-life balance, all those things. And she mentioned something that still today I think about it very, very often, and I tell it to all my students, which is if you really are into science, just do what you think is fun. There's no point of um, spending your scientific career asking questions that you don't consider exciting and fun. That's what we're doing. I mean, science, I think as scientists nowadays, we're really privileged, just to express our curiosity. And having that privilege, take full advantage of it. So, whatever crazy question you might have in your mind, just go for it. Curiosity at the end of the day and curiosity driven research, that's, I think, what makes science move forward with totally unexpected discoveries. And I think this should be nurtured already very early on in your career when you're doing your PhD, that you should, as crazy as possible your ideas might be, go for it. We're here to have as much fun as we can, and frequently it's from those crazy ideas, out of the box ones, that you get the most exciting discoveries ever. So that would be my advice. Go for bold questions, but also thinking that this is your job and you want to have fun while doing it.
Sounds like great advice.
So you're having fun, right, Brenda?
I am. Some days more than others, but I think in general, as the overall balance, I do have fun and I do enjoy. And I did try to go for questions that I think are important or at least exciting. But it's sometimes it feels hard, especially as a early career scientist. There's so much pressure around, getting your next grant fix, getting the yeah, if you're not in a very, positions are have limited terms, or at some point, finding funding for your salary can get a little bit challenging, and that does take a bit of the fun out of it. That you only gravitating to the safe research that will maximize your chances of being financially capable of continuing. And then as you progress and you get a group, and then you have people depending on this for their own, livelihoods, I can imagine that it becomes hard to stay true to that to that motto,
No,
to that
yeah.
role.
I think you're very right. I mean, don't get me wrong when I say that there are no difficulties. Of course, there are difficulties, right? And science in general is becoming more and more competitive. I mean, if I think about the papers I published during my, my PhD, there were 5 or 6 figures, no supplementary figures. And nowadays, to publish in the same set of journals as an immunologist, you have the same number of primary figures, 4, 5, 6, and then 13, 15 supplementary figures. So it's just going insane. And certainly it's more and more competitive. The way we do science nowadays is very different. It's also very much reliant on collaborations within the lab and between different labs, between different institutions. That's really very important. But this mindset, and if you think about it a little bit, it's not like the pressure has to be something very negative, right? Pressure is always there. I think one of the biggest problem at the moment with what you're saying is the modalities of funding in research, right? We're losing a lot these sort of bottom up or curiosity driven type of research. In Europe, we are I think very lucky to have things like the ERC or the European Research Council, which is really a curiosity-driven type of projects and of research, where it's the idea and then obviously all feasibilities, but you don't necessarily need to work specifically on a pathology. You don't need preliminary data. It's all about the originality and the curiosity driven. That's perhaps different in different latitudes, in different countries, but it's really something that I think it would be fantastic to see that happening in other latitudes. To have this kind of funding, and also at a national level, so that at different stages of the career, people could ask these kind of bold questions, rather than thinking in a very, which is the case at the moment, sort of translational type of research, where everything we're doing, especially in biology, has a short-term application for a given disease or pathology, right? And that I would tend to agree with you. That kills it a little bit, to stay true to this idea of go with your most crazy and bold statements in terms of science, right? I do tend to agree, but to encourage, especially the young audience, don't give up, because especially if you select the right lab where to do your PhD, you might end up meeting, a twin soul, a group leader that really wants to do it and has the opportunity to get that type of funding to, do whatever you think of.
All right. Well, with that, I think it's time to wrap up, and thank you for being here. It was a pleasure. It's always awesome getting to talk to someone whose paper we covered very recently.
Yeah, that's true.
Thank you so much, Brandon Jensen. Thank you so much. It was a pleasure.
Yeah, thank you for joining again, Dr. Henrique Veiga-Fernandes from the Champalimaud Foundation in Lisboa, Portugal, for joining to the Immunology Podcast today. That brings us to the end of our show. Don't forget to subscribe to our newsletter at immunologypodcast.com to get the show notes, including episode summaries and links to all of the interview and Roundup Papers. You can also reach out to us on X at Immunopodcast, on Blue Sky, or via email at info@immunologypodcast.com with feedback or to suggest guests. See you next time.